So, I'm Leon Platanes, I'm the director of the Cancer Center, and I want to thank you all for being here. This is the Nat Berlin Lectureship that honors Nat Berlin, who was a pioneer in cancer research and served as the first director of the Cancer Center when it was first founded in 1975 until he retired in 1987. So he was the first one who got funding from the NCI at that point. So Dr. Berlin spent most of his time at the NCI, and he's actually, people don't know this, but he's the one who came up with the concept of physician and scientist. He was the one who came up with the idea and advocated for the concept. And if you look at the history of what was going on then at the NCI, it was very interesting because this had put him at odds with some strong clinical investigators who were pushing for clinical trials. And for some of you, if you read ever Vince DeVita's book on cancer, you will see that there was a big, struggle going on, physician scientists versus clinical investigators those days. So anyway, eventually he left the NCI, he came to Northwestern, and he clearly had made major contributions, and he was the beginning of our cancer center. So that's why we have this lecture. So I want to thank Marcelo and Kathy Green for setting that up. I'm grateful to Michael for joining us today, and Marcelo will introduce Michael.
Thank you, Leon. I am Marcelo Bonini, and I'm a professor in the Division of Hematology Oncology here in the Department of Medicine at Northwestern. And it's my great pleasure this afternoon to introduce Dr. Michael Karin, who is a distinguished professor of pharmacology and the Ben and Wanda Hildyard Chair for Mitochondrial and Metabolic Diseases at the University of California, San Diego. And when I was thinking about how I would introduce Dr. Karin, I think that distinguished in this case really applies from the standpoint that it's very hard to find a scientist that is not only impactful, but transformative to one or two fields. In the case of Dr. Karin, it's actually 3 or 4 or 5. It has been studying inflammation, metabolic disease, cancer biology, transcription factor function, and he has had a tremendous impact in every single one of these areas and also in their interfaces. He has received too many awards to go one by one, but I really would like to mention 2. He has been a member of the US Academy of Sciences for quite some time, and he has received an honorary medical degree from the Technical University of Munich in Germany. His age factor is crazy. It's 256, meaning that he has at least, at the time when this was calculated, over 250 papers that have been cited over 250 times. His laboratory has been very well funded by the NIH, reflecting his impact to many fields. And I think that we are very proud today to have him speak at our annual Berlin lecture. Thank you so much, Dr. Karin. We're looking forward to hear you.
Thank you, Marcelo, for the kind introduction. And I'm really honored to give the Berlin lecture. I read actually about Dr. Nate Berlin contributions, and I understand he was the clinical director of the NCI for many years and so on. So accordingly, I chose a topic of a cancer that we've been working on for quite some time, liver cancer, and a cancer that is actually becoming a bigger and bigger problem every year. So our war on cancer has been quite successful starting about 50 years ago. As you can see, there's been a major reduction in mortality due to many, many cancers. But there are 2 cancers where mortality has been increasing and actually has gone up threefold in the past 40 years. And these are liver and bile duct cancers. And if we will not stop that, they will become the major causes of cancer mortality in the US in the next 20 years or so. So why is that? And part of the reason is that we are seeing a switch in HCC, hepatocellular carcinoma, which is the major form of liver cancer etiology, from virally induced HCC caused by either hepatitis B virus infection or hepatitis C virus infection. We have good vaccines to HBV and very good antivirals for HCV. Actually, the Nobel Prize this year was giving for the development of HCV treatments. But in recent years, in the past 20 years or so, we have seen a switch in HCC etiology from being viral related to being related to the obesity and diabetes epidemic that has been inflicted upon the Western world, especially in the US. And right now, more and more cases of HCC are actually caused by fatty liver disease, NAFLD, and its more acute manifestation, non-alcoholic steatohepatitis. So I will actually focus my talk about NASH-related HCC, because that's the form that we need to understand the pathogenesis, and hopefully come up with useful treatments and, more importantly, preventions of this type of cancer. So NASH-related HCC is actually a part of different causes of HCC, all of which cause the appearance of simple fatty liver disease. And this can be caused not only by consumption of energy high foods such as hamburgers, French fries, or certain soft drinks that contain a lot of fructose, but it's also the result of overindulgence in alcohol. Alcoholic liver disease can also lead to fatty liver disease, and exposure to certain industrial chemicals. All of these causes result in the accumulation of lipids in the liver, and eventually, in some of the patients, lipid accumulation results in inflammation and scarring of the tissue, triggering chronic liver fibrosis that can end up in liver cirrhosis, which in many patients results in loss of liver function. And those who don't die from cirrhosis are at very high risk of progressing to hepatocellular carcinoma within several years. So we've been interested in fatty liver disease and obesity-related liver cancer for quite some time. And we were first attracted to that topic by a very important epidemiological study that appeared in the New England Journal, showing that the incidence of many cancers has grown up with the obesity epidemic, but the major impact of obesity, at least in males, is on liver cancer. So we've been looking for different mechanisms that contribute to liver cancer and contribute to liver cancer in the context of obesity and fatty liver disease. But first, I'd just like to mention our early studies that led to the identification of inflammation, and especially inflammatory induction of interleukin-6 as a major cause of hepatocellular carcinoma development in mice. First, we use a very simple model of HCC induction based on the administration of a chemical carcinogen, diethylnitrosamine, that everybody just thought that it induces cancer by inducing mutations. And it's true that it induces mutations, but we found that even with a mutagen, a very strong mutagen such as DN, tumor development is very much dependent on the induction of liver damage, inflammation, and increase in IL-6 production. And we found that IL-6 can come from different sources, can come from liver macrophages, where it's subject to negative regulation by estrogen receptor activation, thereby explaining why liver cancer is much more common in males than in females, something that we also see in mice. And later on, we have shown that even the initiated hepatocytes or the HCC progenitor cells can express their own IL-6 and responds to the IL-6 in an autocrine manner, leading to activation of JAK1 and 2, activation of transcription factor STAT3, and then progression to full-blown hepatocellular carcinoma. And then a few years later, we became interested in the impact of obesity on liver cancer, showing that there's a 4.5-fold increase in the relative risk of death from liver cancer in obese males with a BMI of higher than 30. We started looking at models where we can see the impact of high-fat diet on the development of HCC. And early on, all we did is combine the injection of diethylnitrosamine with feeding mice with high-fat diet. And we've shown that the high-fat diet consumption actually aggravated the inflammatory response, led to increased expression of both TNF and IL-6. And these were the main factors to which fatty liver or fat accumulation in the liver accelerated the development of chemically induced hepatocellular carcinoma. However, combining DN with high-fat diet is not a very good way to model non-alcoholic steatohepatitis or NASH. NASH is a very complicated disease. It starts as simple steatosis, which is caused by accumulation of lipid droplets in the liver. You can get excessive accumulation of lipids in the liver by taking in too much fat in your diet, but also when you increase lipolysis in adipose tissue, releasing free fatty acids that eventually go into the liver and stimulate triglyceride synthesis. Insulin, another hormone that is induced in response to caloric overload, can also lead to increased triglyceride synthesis. And then when you actually decrease the beta oxidation of fatty acids in mitochondria, you also increase lipid accumulation. But that alone is not sufficient to cause progression to NASH. And that led to the development of the second hit or the multiple hit hypothesis, in which simple steatosis is only the first hit, and then it needs to be accompanied by a variety of secondary hits, including ER stress, mitochondrial dysfunction, and maybe also introduction of endotoxin or high levels of endotoxin into the liver. So to ask whether ER stress has a role in the development of NASH, we had to develop a mouse model in which NASH development is ER stress dependent. Fortunately, we obtained a mouse strain from Eric Sandren, who is currently in Wisconsin, that expresses urokinase plasminogen activator from a liver-specific promoter. And due to overload of the secretory system, the young hepatocytes in these mice undergo ER stress. They suffer some damage, they recover, but when you feed these mice, as opposed to wild-type LEC6 mice that don't express the transgene, you see massive accumulation of lipid droplets, very large lipid droplets. You see ballooning degeneration of hepatocytes, inflammatory infiltrates into the liver, and the development of rather extensive liver fibrosis. And these all together are pretty much the hallmarks of human NASH. And the histological picture of the MUP-UPA liver fat after feeding with a high-fat diet for about 4 months looks very similar or almost identical to that of human NASH. But most importantly for us, being interested in liver cancer, these mice, after 8 months of high-fat diet consumption, will progress spontaneously without any administration of carcinogen to full-blown hepatocellular carcinoma. So we've been using this model to dissect the different mechanisms that lead to the development of NASH and lead to the induction of hepatocellular carcinoma. And just to summarize our earlier results, because I want to focus on more recent findings, we found that ER stress can actually enhance steatosis. Steatosis can cause ER stress, so that's kind of a vicious cycle between ER stress and hepatic steatosis. ER stress can also lead to hepatocyte death, leading to increased production of TNF, which leads to further increase in the synthesis of free fatty acids and cholesterol. Cholesterol has a rather important role in the development of hepatocellular carcinoma. And you also see activation of a variety of inflammatory signaling pathways, including NF-kappa B activation and induction of no other than IL-6, which are previously introduced to you as a very important cytokines that contributes to the development of hepatocellular carcinoma, not only in mice, but now there are many studies that indicate that it has a role in the development of the human disease. More recently, we wanted to see whether other diets that can lead to hepatic steatosis, especially diets that are rich in fructose, whose consumption is increased by about 1,000-fold in the US since the early 50s, can also lead to the development of NASH and to the development of NASH-related HCC. And indeed, we fed the MUP-UPA mice with a fructose-rich diet, and they develop exactly the same manifestations of mice that were given high-fat diet. But this study, whose summary is shown here, allowed us to pinpoint another important factor that contributes to the induction of hepatocellular carcinoma in the context of NASH. And that's deterioration of the intestinal epithelial barrier. And we found that in the case of fructose, formation of the toxic fructose metabolite, fructose 1-phosphate, disrupt the production of certain tight junction proteins by interfering with protein glycosylation. This results in ER stress, increased intestinal permeability, endotoxemia, translocation of endotoxin from the gut to the liver, where it leads to activation of liver macrophages that again produce TNF. And we've shown that TNF not only contributes to the progression from NASH to hepatocellular carcinoma, but it can actually increase de novo lipogenesis and cholesterol biogenesis. Not only in mice, but we also shown that if we incubate normal human hepatocytes with TNF, they don't respond to LPS, they don't express TLR4, but if you incubate them with TNF, you see increased accumulation of lipid droplets in the hepatocytes, and this can be further increased by raising either the glucose concentration or the fructose concentration in which the hepatocytes are cultured, and it's associated with increased activation of SREBP1, increased synthesis of a variety of metabolic enzymes, such as ACLY, ACC1, as well as fatty acid synthase FASN, which accelerate the production of free fatty acids and the formation of lipid droplets. From this, I'd like to switch gears and talk about some of the mutations and the immunological mechanisms that contributes to the progression for NASH to HCC. So we were interested to know whether the tumors that are induced spontaneously in the MUP-UPA mice by either consumption of high-fat diet or fructose actually contain mutations, and do they contain mutations that have also been detected in human hepatocellular carcinoma? So we started by sequencing 16 different HCCs in the mice, and we found that quite a few of them contain mutations such as ARID1A mutations that were found in human HCC, but the sample size was too small, and we could not see a lot of recurrent mutations, and we could not get a real feeling to the mutational spectrum that accompanies the development of mouse HCC. In human HCC, it's much easier because by now people have sequenced hundreds and hundreds of tumors. But what we found, even with this very small panel of sequenced tumors, is that the mutational signature, not the genes that are affected by the mutations, but the mutational signature throughout the genome is very similar between the mouse HCC and human HCC, and you see overabundance of C to T transitions, followed up by C to T transitions, followed up by T to C transitions, and there's a very a much lower incidence of T2A or T2G transversions. Exactly what causes the mutations, we don't know, but we are trying to understand the mutational mechanisms that are actually activated by the appearance of NASH in the liver. But mutations result also in the expression of neoantigens, which can activate the immune system, and that made us take a closer look at the immune microenvironment of the NASH-related HCC in the mice. And very early on, we found that both the mouse-induced, the high-fat diet-induced HCC in the mice, as well as human NASH-related HCC, contain a lot of plasma cells that express immunoglobulin A or IgA, as well as cell-free IgA that accumulates in the liver just the same. And this was also accompanied by appearance of quite a lot of CD8 positive cells. So why is IgA accumulation in the liver or accumulation of IgA producing plasma cells interesting? And the reason that we found it interesting is that it was described already 40 years ago that IgA accumulates in a variety of liver diseases that are associated with liver fibrosis. And more recently, the group of Quentin Ansey at Newcastle have shown that serum immunoglobulin levels, especially IgA, can predict progression to fibrosis in patients with non-alcoholic fatty liver disease. So we decided to take a closer look at these IgA producing cells, And actually, we started working on these cells quite a few years before we detected them in the liver. And we found that in the context of prostate cancer, these cells acquire the ability to produce PD-ligand 1 and thereby being able to induce the exhaustion or the dysfunctionality of cytotoxic T cells, CD8 positive CTLs. And in addition to PD-L1, they also produce very high levels of IL-10, which also contributes to the dysfunctionality of the T cells. And the reason these cells form is that when you have liver fibrosis or increased activation of cancer-associated fibroblasts, CAFs, in the case of prostate cancer, you get increased production of TGF-beta. There's also increase in the production of IL-21, and both of these cytokines induce the IgA class switch recombination that converts naive B cells to IgA producing plasmablast and plasma cells that also acquire PD-L1 and IL-10 expression. So we wanted to see what happens in the mouse model. And as I mentioned to you, there's extensive accumulation of IgA positive cells in the MUP-UPA mouse after consumption of high-fat diet. But we found that any form of liver fibrosis, whether it results in the appearance of HCC or not, results in the accumulation of IgA positive cells in the liver. These cells mostly have the morphology of plasma cells. Some of them are for earlier developmental stage, plasmablasts, but they are very much the same cells that I just described to you in the previous slide. But what is the importance of these cells in the development of hepatocellular carcinoma? So to ask these questions, we have generated MUP-UPA mice that are also deficient in the expression of IgA, a mutation in the IgA locus. And we found that when we knocked out IgA, we slowed down tumor development and decreased both tumor multiplicity and tumor size, resulting in a major decrease in tumor burden. And since we postulated that the IgA positive cells promote HCC development by inducing the dysfunctionality or the exhaustion of CD8 positive T cells, we also generated a MUP-UPA mouse that was deficient in CD8 positive cells. And then, to much of our surprise, at least my surprise, there was huge acceleration in the development of HCC. We found that about 25% to 30% of the mice already had very big tumors at 6 months of age, at a point where we normally don't see any tumors in the MUP-UPA mouse. And some mice also had tumors already at 3 months of age. And then if we waited the entire 8 months to 9 months complement of high-fat diet feeding, we just got a liver that was just composed of tumors with very little normal tissue. So why is that? And it turns out that indeed, as we speculated, when we knock out IgA in the liver, we increase the number of effector CD8 positive cells, both before tumor appearance, when the mice only have NASH, and also the presence of these cells within the small tumors that appear in the IgA negative mice. We look at a variety of exhaustion markers, and it was very clear that in the absence of IgA, we were not inducing CD8 exhaustion, but in the presence of IgA, many of the CD8 positive cells that we found in the liver were actually exhausted. So since PD-L1, PD-1 signaling can lead to CD8 cell exhaustion, we ask what would happen when we knock out both the PD-L1 and PD-L2 genes and preventing the induction of T cell exhaustion. And what we saw is very similar to what we saw in the IgA knockouts. The PD-L1 deficient mice also developed very few tumors, and the tumors were much smaller. But here we actually found something else that was interesting. We found that in addition to inducing the exhaustion of CD8 positive cells, has been known for quite some time, PD-L1 and 2 were also important for the appearance and the class switch recombination that leads to the development of IgA positive cells. And you can see that knocking out PD-L1 and 2 really reduces formation of the cells by almost 50-fold, not as much as knockout of TGF-beta receptor 2, but quite a substantial reduction in formation of these cells. So we wanted to know whether the CD8 positive cells are really responsible for tumor regression, and if so, are they really recognizing tumor-specific antigens? So what we did is we isolated CD8 positive cells, both from the spleens of the HCC-bearing mice and from the tumors, sequenced their TCR alpha and beta chains, and found that where in the spleen, the TCR repertoire, this is just shown for TCR alpha, is very complex. There are tens of thousands different TCR alpha sequences that you see in the spleen, but within the HCC, there is clonal expansion of only a small number of these TCRs. So the response is really oligoclonal, suggesting that these are the T cells that are activated and expanded in response to expression of tumor-specific antigens. We have not cloned the tumor-specific TCRs as yet, and we haven't been able to identify the tumor antigens that activate them, but we hope to do so in the future, as a lot of this technology is being developed right now. But all of this led us to predict that maybe anti-PD-1, PD-L1 therapy would be effective for hepatocellular carcinoma, and following its success in melanoma and non-small cell lung cancers. And early on, it was suggested that the reason that melanomas and non-small cell lung cancer respond so well to inhibition of the PD-1 checkpoint is because they have very high mutational burden, and there's very extensive accumulation of tumor-specific CD8 positive cells in such tumors. However, the mutational burden in hepatocellular carcinoma is much lower, but we decided to test, because we saw accumulation of PD-L1 in the NASH-inflicted liver, what would happen when we treat them with an anti-PD-L1 antibody that we obtained from Ira Mellman in Genentech. And indeed, just like the knockout of PD-L1 and 2, we found that anti-PD-L1 monotherapy in these mice was very effective. It got rid of about 60 to 70% of the tumors. The residual tumors were very small. And the only few cases where we did not see a good response to anti-PD-L1 was in the case of tumors that were surrounded by very extensive peritumoral fibrosis, which we believe prevents the access of the activated T cells into the tumor. And pretty much at the same time when we completed these mouse experiments, turns out that somebody did the clinical trial in HCC despite the disbelief that HCC will respond to immunotherapy. And they found that indeed treatment with an anti-PD1 antibody, this is an antibody produced by Bristol-Myers Squibb. But since then, many anti-PD1 antibodies produced by Merck, AstraZeneca, were all found to be effective. And what you see in this paper that just came out at about the same time that our mouse paper has been published is that there's about 15 to 20% response rate. The response is quite durable, much more substantial than the only targeted therapeutic that was approved for HCC treatment, sorafenib, a PAM kinase inhibitor, accomplished, which is about 3% response rate, and only about 2 or 3 months increase in survival. Here, the response was rather extensive and quite durable. Since then, people have, or different companies have started testing the combination of anti-PD1, PD-L1 antibodies with other therapeutics. And it's been observed now at least 3 times that different VEGF receptor inhibitors, all the way from small molecules to Avastin, dramatically increase the response rate to PD-1, PD-L1 blockade in hepatocellular carcinoma, resulting in response rates that exceed 40%. So this really revolutionized the treatment of hepatocellular carcinoma, a tumor type that really had no treatment other than partial hepatectomy or complete removal of the liver and transplantation of a new liver. Again, in support of the immune mechanisms that really drive the development of hepatocellular carcinoma, we found that unlike many tumors in which have been selected to show reduced expression of MHC1 antigens and thereby avoid the activation and of CD8 positive cells, expression of MHC1 antigens is quite high in hepatocellular carcinoma. These antigens will, MHC molecules will produce HCC-related neoantigens to CD8 positive cells, lead to their activation. But when these cells reach the liver that contains a lot of PD-L1 producing IgA positive cells, due to the chronic inflammation and fibrosis in the liver, they will shut down the activity of the cytotoxic T cells and inhibit their ability to control the development of hepatocellular carcinoma. So this immune mechanism is quite important, but it's not the only mechanism that contributes to the development of hepatocellular carcinoma. So I'd like to take you back to some of our earlier study that show that there's also a very important role for metabolic pathways, and especially metabolic switch that I will present to you in the development of HCC. So what we found early on in the MUP-UPA mouse is that in response to consumption of high-fat diet, these mice accumulate very large amounts of a protein called P62. And it appears already before they progress to hepatocellular carcinoma. So what is P62? P62 is an adapter protein that normally has a role in the shuttling of polyubiquitinated protein aggregates and organelles, such as damaged mitochondria, to autophagosomes and leading to their elimination by autophagy. And it's been observed by quite a few groups, including Gökhan Hotomisligil before us, that stress and consumption of high-fat diet can perturb autophagy in the liver. And that's probably one of the main causes of increased P62 accumulation. The other increasing fact about P62 is that it accumulates in NASH and in the hepatocellular carcinoma in about 60% of them through formation of what's called Mallory-Denk bodies, which is a pathological hallmark of both NASH and NASH-related HCC. And we found that if we knocked out the P62 encoded by the sequestrin-1 locus in the MUP-UPA mouse, we really reduced and attenuated the development of hepatocellular carcinoma without much effect on the accumulation of lipid droplets in the liver. So we ask, how does P62 manifest its oncogenic effect? And to make a long story short, we found that P62 accumulation can lead to activation of mTORC1, but more importantly, it led to activation of a transcription factor that plays a very important role in the antioxidant defense response, NRF2. And NRF2 was found to be activated mutationally in 9 to 12% of hepatocellular carcinoma. This is associated with either gain-of-function mutations in the NFE2 L2 gene that encodes NRF2, making NRF2 resistant to the binding of KEAP1, a protein that targets it to ubiquitin-dependent degradation, or loss of function mutations in the KEAP1 locus that prevent the expression of functional KEAP1. Then NRF2 will accumulate and would lead to activation of genes that protects cells from oxidative stress, from different oxidants, and can somehow also promote the synthesis of glutathione and promote metabolic reprogramming that leads to the development of hepatocellular carcinoma. But this is what we thought earlier on. So we thought that inhibition of cell death due to oxidative stress was very important. Turns out we were probably wrong. And the more important function of NRF2 in HCC development is to cause a metabolic switch that completely perturbs liver metabolism to be much favorable to the formation of hepatocellular carcinoma. So we started asking, does NRF2 activation play an initiating role in the appearance of HCC, or does it play only an auxiliary role in the development of HCC? In addition to HCC, NRF2 activating mutations are extremely common in a variety of squamous cell carcinomas, both in non-small cell lung cancer, head and neck cancer, and so on. And in rat models of HCC, they were found to appear very, very early, suggesting that they may contribute to tumor initiation. But to know exactly what they do in the mouse, and by extension in the humans, we generated a mouse with conditional activation of an NRF2 allele that codes for a protein that is refractory to the binding of KEAP1 and is resistant to KEAP1-induced degradation. This is actually a mutant allele, first isolated from lung cancer. And we can control the expression of this allele by a LOX-P stop cassette. We can excise it with different Cre recombinases. So in this case, we use the albumin Cre to activate it in the liver. And instead of formation of liver cancer, at least for about 1 year, for which we followed the mouse, we found that NRF2 activation leads to very rapid liver enlargement or hepatomegaly. However, this hepatomegaly was not due to increased number of hepatocytes, was not due to hyperplasia, but was actually due to hypertrophy. Turns out that the hepatocytes in these livers are much larger, mainly due to the accumulation of glycogen, something that is not normally seen in the mouse liver. And it's really extensive. In 8 weeks old mice, you really see the hepatocytes just bursting with glycogen deposits. We had other ways to activate NRF2, and it all resulted in the same phenotype. So we conducted RNA-seq analysis on these livers to fully understand what's going on metabolically, why we have increased glycogen formation. And we found that a lot of genes that are involved in glycogen synthesis and also gluconeogenesis were upregulated in these livers. But we also found that genes that are associated with activation of the G2M checkpoints were upregulated when we activated NRF2, either by overexpression of P62 or in this transgenic mouse model. That explains why we are not seeing hepatocyte proliferation. We are just seeing hypertrophy because all of these genes contribute to a G2 arrest, which we confirmed in these livers. But we also saw massive upregulation of genes involved in fatty acid metabolism, again, explaining why we see increased accumulation of triglycerides in these livers. But as I said, we also found that NRF2 activation exerted many effects on metabolism that we surprisingly found to depend on the activation of AKT. And when we treated the mice with AKT inhibitors, we completely prevented the development of hepatomegaly, but we could not understand how NRF2, which is a transcription factor, leads to activation of AKT. But eventually, what we found through the RNA-seq analysis, that activation of NRF2 either by overexpression of P62 or expression of this degradation resistant allele in the liver resulted in upregulation of many TGF family members, including TGF-alpha, ARAG, EGF, and also effects on certain members of the PDGF family. And we found that in these livers, we saw increased activation of EGF receptor, thereby explaining the activation of AKT and the dependence of the disease development on AKT activation. And eventually, we confirmed that human hepatomegaly is also dependent on EGF receptor activation and AKT activation. But the question is, what is the role of NRF2 in the development of hepatocellular carcinoma? And as I said, when we activated NRF2 alone, we did not see any cancer. So we decided to go back to the MUP-UPA model and introduce this activated NRF2 allele into the MUP-UPA mice, activate it in hepatocytes, and then follow the mice kept either on normal chow or high-fat diet. And whereas MUP-UPA mice with activated NRF2, again, did not develop any hepatocellular carcinomas, they only show some benign liver tumors. In response to feeding with high-fat diet, we got very rapid development of HCC. And as I shown you before, we normally have to wait 18 until the mice are about 10 to 11 months old to see formation of hepatocellular carcinoma in this model. But in this case, we start feeding the mice when they are 8 weeks old, just after weaning. And after only 4 months of exposure to high-fat diet, we get rather extensive development of hepatocellular carcinomas. Again, some of the cells in these HCCs show accumulation of glycogen as indicated by this glassy appearance. So we ask, what is the effect of NRF2 activation on liver metabolism that may be relevant to the development of hepatocellular carcinoma? And as I mentioned earlier on, we saw again that activation of NRF2 in the MUP-UPA context results in AKT activation, which is due to increased production of growth factors. We got increased activation of a variety of lipogenic enzymes controlled either by SREBP1 and responsible for de novo lipogenesis, ACLY, ACC1, and fatty acid synthase. And we also see upregulation of HMG-CoA synthase and HMG-CoA reductase that are responsible for cholesterol biogenesis. But what really caught our eye is the degradation of this protein FBP1, which almost completely disappeared in response to NRF2 activation. So what is FBP1? FBP1 is an enzyme, fructose 1,6-bisphosphate phosphatase, which is responsible for reversal of glycolysis to gluconeogenesis. So FBP1 catalyzes the opposite reaction to phosphofructokinase, and it converts fructose 1,6-phosphate to fructose 6-phosphate, and that also accelerates exosamine synthesis. It may lead to increased production of NADPH through the pentose phosphate pathway, but it also has quite a few other effects on liver metabolism that I would like to share with you. But first, I would like to focus what is the role of FBP1 in hepatocellular carcinoma and how NRF2 regulates FBP1. So earlier this year, Celeste Simon, who started studying FBP1 long before we did, has shown that FBP1 can act as an HCC tumor suppressor. And she suggested that in the absence of FBP1, which in her case, she just induced by knocking out the gene, but it has been observed that HCC development is also associated with FBP loss, results in the induction of ER stress because it interferes with exosamine synthesis, and that leads to activation of hepatic stellate cells that then feed in to support the growth of hepatocellular carcinoma. We think that FBP1 operates cell autonomously through an entirely different mechanism. But we did obtain a lot of the reagents from Celeste with whom we collaborate. So we ask first, is the degradation of FBP1 unique to the mouse with the genetic activation of NRF2? Or can we knock out ATG7 in the liver, disrupt autophagy, accumulate P62, which leads to activation of NRF2, and would that lead to degradation of FBP1? And indeed, in the ATG7 deficient mouse, you see very strong accumulation of NRF2 and FBP1 degradation. These mice do not develop HCC, they actually develop hepatic adenomas, but we see all of this even before the hepatic adenomas appear in these mice. And in the MUP-UPA mice, we see that in certain mice, there's downregulation of FBP1, but it already appears in the non-tumor tissue, and it's not much more extensive in the non-tumor tissue than in the tumor. So we think that maybe the downregulation of FBP1 actually precedes HCC development, and it makes the liver metabolic landscape much more permissive to HCC. When you downregulate FBP1, you upregulate glycolysis. This can definitely contribute to the glycolytic shift that is seen in many cancers, and it also upregulates lipogenesis. But the question is, how does NRF2 induce FBP1 downregulation? And we found that the activation of NRF2, because it leads to activation of AKT and as well as MAP kinases, can support the degradation of FBP1 through activation of either MAC or ERK. Inhibition of AKT had no effect on the degradation of FBP1, but ERK and MAC inhibition completely block the degradation of FBP1, not only in the mouse hepatocytes, but also in 2 human hepatocellular carcinoma cell lines, HepG2 and HUH7. Eventually, what we found is that the phosphorylation of FBP1 by ERK2, which is activated in response to increased EGF production, and we can also activate it by increased expression of a gain of function mutant in the ERK2 gene, induces the ubiquitination of NRF2 because it increases the recruitment of a ubiquitin ligase called TRIM28 that was also found to be upregulated in hepatocellular carcinoma and was already shown to promote the Warburg effect, as we suggest, by targeting FBP1 for degradation. But exactly how this pathway is activated was not known. So we now can show that it's really activated in response to NRF2 activation. You get extensive ubiquitination of FBP1, which is then targeted for proteasomal degradation. But quite surprisingly, we found that if we overexpress FBP1 in cells with upregulation of NRF2, FBP1 overexpression also led to the degradation of NRF2. So not only NRF2 promotes the degradation of FBP1, FBP1 promotes the degradation of NRF2, and again, it's due to NRF2 ubiquitination. In this case, it's KEAP1 independent. It depends on phosphorylation by GSK3 beta, glycogen synthase kinase beta. But exactly how FBP1 affects the activity of GSK3 beta, we still don't understand. But importantly, by controlling the level of NRF2 expression and FBP1 expression in 2 human HCC cell lines, we can either increase or slow down their proliferation in vitro. We still have now to check the effect on tumor formation in the mouse system and the tumorigenic activity of these cell lines. But you can see that if we introduce into these cells the activated form of NRF2, they proliferate much faster, they become much more viable. If we introduce into them FBP1, we really slow down cell proliferation quite extensively. And if we overexpress FBP1 together with the activated form of NRF2, looks like FBP1 is dominant. And again, it slows down cell proliferation in both of these cell lines. So what we think is going on in the development of NASH-related HCC, in addition to activation of NF-kappa B, TNF signaling, immunosuppression through IgA production, and so on, there's also a very important metabolic switch that consists of NRF2 and FBP1. NRF2 activation would lead to downregulation of FBP1. This will support the Warburg effect, increasing glycolysis, enhancing cell proliferation, and also enhancing lipogenesis. FBP1, on the other hand, we think is indeed a tumor suppressor in the liver, not because it prevents the activation of ER stress, but because it induces the degradation of NRF2 and switching glucose metabolism to gluconeogenesis instead of glycolysis, and slowing down tumor metabolism and preventing tumor development. And that's something that we hope to understand in the future. And still quite a few other things to understand, which I mentioned during my talk. And finally, I just like to acknowledge all past and present lab members who contribute to this work. The list is too long to go through each one of them, but I'd like to point out Yelena Todorich, who actually extended the work of Hayato Nakagawa and Atsushi Umemura, who established the MUP-UPA NASH model for HCC induction. She's shown that it also works for fructose rich diet. Shabnam Shalapur, who left the lab a few months ago to start her own lab at MD Anderson, did all the work on the liver immune microenvironment. And Feng He generated the mice with the activated NRF2, figured out that it controls glycogen metabolism. And Li Gu is responsible for identifying this crosstalk, very important switch between NRF2 and FBP1, something that we hope to complete in the very near future. We had many collaborators who assisted us in this work. And I also like to thank you for your attention, and I'll be happy to entertain questions.
Thank you so much, Dr. Karin. I would ask you just if you could stop sharing your screen so I can present you with a commemoration of your talk here. I will start sharing mine.
Okay.
This is, I don't know if you guys can see it. I think you can. So this is the Dr. Nathaniel Berlin, after which this lectureship is named. And I would like to virtually present Dr. Karin with a commemorative gift of his talk here at the Lurie. We are very grateful for your talk. And I do have 3 questions for you. I think that's as much as we will abuse you. So starting with Team Lane, if you could please unmute and ask your question. team? Well, so we'll go for the second question. Bill Miller has a question.
Yes, hi, great lecture, lots to think about. My question is naive. It won't be nearly as thought-provoking as Navdeep will be, as Navs will be. I want to go back to the MUP-UPA model. And I just don't understand, maybe you can explain it. So these are mice that overexpress UPA in their hepatocytes. It causes ER stress, of course, but some of it has to be leaking out. And so UPA is going to activate plasminogen to plasmin and set off a whole lot of other pro-inflammatory cascades, as well as doing things that tumors like to grow, like digesting extracellular matrix. So I'm wondering, is the UPA that these mice make non-functional, or how do we account for the contribution of the UPA to the inflammatory environment?
Okay, that's actually not a naive question. It's a very good question. And I've been asked that many times. And we actually were promoted to look at the receptor for UPA. And what we did is we actually looked at older mice that developed NASH and then developed HCC. And we did not see any effect for activation of the protease receptor. And so we don't think that at that stage, UPA is functioning as UPA. Also, these mice express very high levels of UPA about 5 to 6 weeks after birth, and then it starts going down. So at 6 weeks of age, they really have a lot of ER stress in the liver, and the liver is partially inflamed, as you said. And part of it could be to the UPA. But then it goes down and we cannot detect it. But then when we give them high-fat diet, they are somehow much more prone to development of ER stress than the black 6 mice. Maybe they start expressing low amounts of UPA and that's sufficient. But to test whether overexpression of any secreted or poorly folded protein in the liver can lead to that, we collaborated with Randy Johnson at the Burnham Institute. And Randy has been working on factor 8 and factor 10 for many years. And finally, they managed to express different forms of factor 8 through hydrodynamic injection. And the ones that fold and get secreted properly and does not lead to activation of the UPR, does not lead to ER stress, is not oncogenic. But it's enough to overexpress a poorly folded protein, such as factor 8, and that would also lead to induction of HCC in mice. So we think it's really the ER stress in combination with the high-fat diet and so on that plays the major role.
Yeah. Related to that, the experiment of nature, people with alpha-1 antitrypsin deficiency get cirrhosis, and they are also more likely to get hepatocellular carcinoma. I don't know if anyone's studied the ER stress in that, but well, it would make sense.
Yeah. I mean, that's a good question, but it's clear that alpha-1 antitrypsin accumulation does exert its pathogenic effect through the induction of ER stress. But exactly what's going on in the experiment of nature, as you mentioned, leading to hepatocellular carcinoma, we don't know. But, you know, any kind of disease that damages the liver over extended lengths of time increases the risk of fatty liver, NASH, and liver inflammation and hepatocellular carcinoma. So it's not only ER stress, but ER stress is definitely a major pathogenic factor.
Yeah, thank you.
Thank you. Nav?
Michael, great talk. Just a quick question. By the way, it's not Randy Johnson who gave you that. It's Randy Kaufman, right?
Oh, yeah, Randy Kaufman. Sorry, don't even mention it to Randy.
Yeah. So, you know, there's these NRF2 activators that people are using potentially as chemopreventative agents, as you know. And probably the most famous one from the NRF2 activators might be DMF, which is used for psoriasis.
Yeah.
That just started about only 7-8 years ago. But so, I mean, one concern would be the long-term use of something like DMF, which is an NRF activator, potentially might make you more susceptible to liver cancer as you age 20 years, 30 years of using that drug, no?
Yeah, that's a good question. So the question is, how strong an activator is DMF? The one I'm more worried about is disulfiram, that is actually introduced by certain people as a chemopreventive agent that reverses the activity of aflatoxin that can lead to HCC development. So I think, you know, early activation of NRF2 is protective, and it will accelerate the metabolism and the inactivation of different xenobiotics. So it protects the liver. But when it goes on forever and ever, it completely alters the liver metabolism, and it can lead to cancer, and even before cancer, to metabolic disease. So I think it remains to be seen how long can we treat people with these compounds, and how safe they are. I can tell you that we also been studying NRF2 in pancreatic cancer, and we can make organoids that only have ROS activation, so they are not tumorogenic. We treat them with H2O2 for 5 days. We activate NRF2, and they become completely tumorogenic. So we can get this whole switch now in vitro. And the H2O2 does not work through mutation. It actually works through NRF2 activation. So we are actually doing an experiment now with disulfiram to see if it will induce the tumorogenicity of KRAS-initiated cells in the pancreas. But there's an ongoing battle between the people who think NRF2 is protective and the cancer community who recognize that NRF2 is oncogenic long time ago. First in lung cancer, and then in pancreatic cancer, as David Tuveson did, and so on. So you cannot deny the effect of the NRF2 activating mutations.
Right. Thank you. Good seeing you.
Fantastic.
Thank you.
Well, I'd like to thank everyone, and I'd like to thank Dr. Karin for giving the talk today. Really appreciate it. It has been a pleasure to have you here, and I'm sure everyone learned a lot. Thank you so much.
Thank you. Thank you. And I'm waiting for your...
Oh,